So thank you for dialing in to this this evening. As previously advertised, I'll be talking to you about challenges at mRNA COVID vaccine development and manufacture. And as I said, I'm a chief technologist at CPI and have spent the last 18 months doing a lot of work on mRNA. So in terms of the content, we'll have a little brief refresher on the background to vaccines. What has needed to be done to make mRNA a vaccine? There's quite a lot of work by a number of people over a number of years has gone into getting us in a position where mRNA can be used as a vaccine. Something about the features of mRNA as a vaccine that makes it so very attractive and indicates great promise to the future. Then go on to talk about how you'd make an mRNA vaccine or an mRNA at scale. Talk then briefly about UK's capability of manufacturing mRNA, and then just some conclusions. So first of all, a little bit of background to CPI. So CPI, it stands for the Centre for Process Innovation. We've been in existence overall for about 15 years now. We're part of the UK's high value manufacturing catapult, so part of the catapult network. You've probably heard of the cell and gene therapy catapult, and we're part of that catapult network. And our remit is to support the development of advanced manufacturing in the UK, helping people with innovative ideas and innovative processes, and helping them take these ideas through the difficulty of scaling up and making sufficient materials to help develop markets to get them to a point where they can then, it's self-sustainable and they can go off and make materials, they can go off and sell their products. And we're based in the northeast of England. We've got a number of sites. So I myself operate from our Darlington site where the National Biologics Centre is. We have a site at Wilton, which is in industrial biotechnology. A site at Sedgefield, which has 2 units, one of which is the looking at formulations, one is looking at printable electronics. At Newton Aycliffe, we have a pilot plant for manufacturing printable electronics. And in Glasgow, we're in the process of constructing the Medicines Manufacturing Innovation Centre, which is looking to improve and revolutionise the way we make small molecule pharmaceuticals. So looking at things like continuous manufacture of small molecule pharmaceuticals, looking at changing dosage forms on the fly, all sorts of innovative technologies for making small molecule pharmaceuticals more advanced or more efficiently. In terms of the biologics facility in Darlington, it's 5,000 square metre facility. It was construction was completed in 2016, and that then cost around just under £40 million. We've got comprehensive expertise in microbial and mammalian expression technologies, and that's based on a depth of experience supported by the up-to-date facilities we have. Direct experience has included monoclonal antibodies, viral vectors. We've done a lot of work on adeno-associated virus, AAV, and lentivirus, maybe less so on adenovirus. We've worked on a large number of non-antibody proteins from mammalian and microbial sources, worked on virus-like particles, and had a lot of experience in conjugation technologies where chemistry is combined with biology to make things like antibody drug conjugates and half-life extension of proteins by binding things like molecules like polyethylene glycol to them. We've done work on nanomedicines, which includes mRNA and its encapsulation. And we'll clearly talk a lot more about that. We've got 13 flexible laboratories designed to handle organism up to BSL-2. We worked overall to an ISO 9001 quality system, and at the moment a number of suites are being modified for GMP operation. We've found that there's a lot of demand for early phase GMP operation, good manufacturing practice for making early phase clinical material, and that should be available in late 2021, later this year. So moving on to vaccines, some little bit of a very basic refresher. And so a vaccine primes the immune system to recognize an antigen, which allows the immune system to respond more rapidly to subsequent presentation of the antigen to the organism. So that's the antigen is typically from a pathogenic organism. In COVID-19, it's from the COVID virus itself. And typically we think of, if you look on the right-hand side here, we've got, I guess, the basis of all biology. It's you start with DNA encodes, sequences are then translated into RNA, which is then converted into protein. And it's typically been protein products in their broadest sense that have formed the basis of all antigens in classical vaccines, or vaccines to date very recently. So typically antigens have been delivered in the past in the final form to the patient. So say we've got classical vaccines which use live, attenuated or non-viable cells, and there's still lots of vaccines out there like this. So there's polio is a classic example. I think there's vaccines to measles and mumps and rubella. You've got conjugated vaccines where glycans are conjugated chemically to immunogenic proteins like CRIM197, and vaccines like that are available for pertussis, and you may have heard of Prevnar 13 for childhood diseases. Virus-like particles typically produced in recombinant yeasts, Pichia pastoris, Saccharomyces cerevisiae being very common manufacturing platforms. VLP vaccines have appeared more recently. So the HPV vaccine is a virus-like particle vaccine. It's made in, depending on whose HPV vaccine you take, it's either made in Saccharomyces cerevisiae or it's in a baculovirus. And subunit vaccines, where the key proteins displayed by the disease organism are provided as the antigenic species, and things like that would be tetanus toxin, and in the past I've worked a little bit on biodefense vaccines. Things like vaccines to anthrax or to plague would fall within the subunit vaccine category. In the last few years, there's been interest in having the antigen produced by the body, and this is very similar to gene therapy in that you deliver either DNA, which is the, I guess, the more established technology, or RNA to the body. So delivering DNA in the form of plasmids or viral gene therapy, typically through adenovirus, is a way of having the body produce its own antigens. These are still very recent vaccines. The first of these would be one for Ebola that was only licensed in 2019. And then much more recently, the mRNA vaccines, which moved down the supply chain, where you're formulating these into liposomes and polyplexes. And the first of these really only saw human use from late last year onwards. And yeah, something about COVID vaccines themselves. All the vaccines that people have been looking at have been derived from the coronavirus spike protein, and this is the coronavirus as shown here. It's a single-stranded polymerase RNA virus, around about 30 kilobases in length. There's a 5' cap, and we'll talk more about 5' caps. And we've got a 3' poly A tail, again, indicating some of the things that you might want to consider when making an mRNA vaccine. The spike protein, which is covers the virus surface, it binds to angiotensin converting enzyme 2, ACE2, and which parts of the spike protein interact with which parts of ACE2 are really quite well identified. And that interaction is responsible for cell entry. The spike protein binds to ACE2 and allows the virus to enter the cell. And there's a few known mutations. This lysine mutation, this glutamate mutation, this arginine mutation. I've forgotten exactly which ones, but one of these is associated with what was one of the original mutations, the Kent mutation, and then we've got the South African mutation here and here, and the Delta variants follow the same course. I think they're round about later on in the sequence, or at higher numbers in the sequence. So as you can see, the spike protein is the thing that most of the vaccines are designed to produce, and it's no different for mRNA. In terms of mRNA COVID vaccines at the moment, there are 2 approved vaccines. The vaccine made by Moderna and by Pfizer/BioNTech. And they're both mRNA vaccines with modified nucleosides. And it says these are in phase 4, so they've been approved for use, and they're being post-marketing monitoring going on just now. There's another 2 vaccines that are still at clinical stages. So there's an mRNA vaccine made by CureVac, which is at phase 3. It's using non-modified nucleotides. And one from Arcturus Therapeutics, which is a self-amplifying RNA vaccine at an earlier phase 2. So we've got two approved vaccines, 2 in clinical trials from mRNA, and another 24 in preclinical trials. And there's data from the WHO, which also indicates there's probably about 170 vaccines at the moment in preclinical trials. So mRNA is making up quite a big chunk of these. And why the interest in mRNA vaccines? Well, they're controllable. mRNA is non-infectious in its non-integrating platform. One of the challenges of working with mRNA is it's extremely easy to degrade, and that's one of its advantages. It's degraded by normal cellular processes. It doesn't build up to any extent, but it does present a lot of challenges during manufacture of vaccines or mRNA for any purpose at all. Its in vivo half-life can be regulated through the use of various modifications and delivery methods, which we'll come on to. It's a minimal genetic vector, and anti-vector immunity is avoided. So for viral vectors, you might be able to develop immunity due to repeated presentations of the viral vector. That's not going to be the case with mRNA. It's rapidly deployable. You need only very small amounts to be potent. The BioNTech vaccine is only uses 30 micrograms per dose, a very small amount. Okay, there's 2 doses, so 60 micrograms in total, but that's still a small amount to manufacture. It's compact manufacturing used to the high yield of the transcription processes used to make RNA. There's no cell line development required, so you don't have the complexity of going off and developing a clonal cell line, which takes time and effort, and then doing that to GMP. It's a platformal process in that it's probably going to be independent of the size or the sequence of the antigen being expressed. And now we've got real-world validation. There's several hundred millions of people have received an mRNA vaccine. In terms of making this, there are three components, which is worth reflecting on. The first of these is the template that determines the sequence of the mRNA, and this is typically from a DNA plasmid produced in E. coli. It needs to be highly purified, remove the host cell components, definitely remove any RNases coming from the host cells, and the quality of the template directly affects the mRNA quality. So you make a plasmid, but then you need to do some linearization for mRNA production. And there's some other alternatives to plasmids being considered, such as rolling circle DNA amplification. Having made your linearized DNA, the template is then used in an in vitro transcription reaction, which is an enzyme reaction using an RNA polymerase, where it's a one-pot enzyme reaction where you just throw in the nucleases and generate your mRNA. It's very highly productive. You generate about 5 grams per liter in a 2 reaction of mRNA, and then you purify the reaction components away from the mRNA you've made to be able to do encapsulation. And that's where the mRNA you've made is encapsulated in the lipid nanoparticle. This is extremely important for being able to use mRNA as a in vivo. It's essentially required to protect the mRNA from endogenous RNase activity during administration. It allows also the mRNA to enter cells and be transcribed. And it's a complex lipid mixture required in making a nanoparticle. And understanding how to make these and the components required to make them has certainly been one of the things that have been essential for making mRNA as a product. Okay, move on to the mRNA itself. There's 2 basic flavors, and we'll just look at some of the design elements just now. First of all, consider mRNA or modified RNA, mRNA. They both have this sort of structure here where there's a cap required to reduce recognition by the host defense systems to viruses, or sorry, the host defense systems to non-host mRNA. It has a 5' untranslated UTR region to maximize antigen expression, a 3' UTR region to also help with antigen expression, and a polyadenosine tail to, again, increase half-life in the body. If you've done that, your mRNA is translated and you generate an antigen from the mRNA sequence. Some things that have been in mod RNA, you would swap out uridines for methyl pseudouridine, which increases the half-life again and has some advantages in terms of increasing the levels of antigen expression. And you just do this by swapping out, when you're doing the in vitro transcription reaction, you swap out uridine for methyl pseudouridine in the mix you're using in the IVT reaction. And for COVID-19 vaccine, the size is around about 5,000 nucleotides. The spike protein is quite large. It's around about 1,200 amino acids. Moving on to self-amplifying RNA. These are based on viral sequences. So as well as the capping structure and the 5' UTR and the 3' UTR, they also contain 4 proteins from a viral sequence, which when expressed generate a replicase, which copies the mRNA and makes further copies of itself. There's quite a cunning way, so you make multiple copies of the mRNA you've dosed into the subject, and then goes off using the subgenomic promoter to generate multiple copies of the antigen. Otherwise, everything else is about the same. You've got the cap, the 5' UTR, the 3' UTR, and a poly A tail to increase half-life. Because of these non-structural proteins, the saRNA is quite a bit longer, quite a bit larger rather, around about 12,000 nucleosides in the COVID saRNA vaccines. And the advantage of self-amplifying RNA is that you don't require as much of it to generate enough antigen to be a viable vaccine. So the mRNA doses are lower than the 30 micrograms that would be with the conventional modified mRNA. In terms of making the mRNA itself, it's touched on. It's a one-pot enzymic synthesis. You have the DNA template, which is a linearized plasmid. You've got some key enzymes that you'd add together. So we've been typically using the T7 RNA polymerase from the T7 phage. There's a pyrophosphatase added because the reaction generates a lot of pyrophosphate, and if you do that, you'll just have a lot of insoluble pyrophosphate. Having the pyrophosphatase there converts it into soluble phosphate. As always with RNA work, you have an RNase inhibitor, and at the completion of synthesis, you'd add DNAs to digest up the template. And the polymerase moves through binds initially to the promoter regions on the template, and then moves in a transcription bubble through the template with mRNA being generated as shown. You add equal amounts of all the nucleotides, and as very important, we'll come on to the RNA capping reagent, which is essential for minimizing activation of the host immune system. And in about 2 hours at 37 degrees, you'll generate about 5 grams of mRNA following the sort of protocol, which is one of the major plus points with an mRNA vaccine. It's very high yielding. It's very quick to produce mRNAs. If we then come on to the T7 polymerase itself, its X-ray structure was solved over 20 years ago, and by Tom Steitz's group. They looked at the structure with a DNA sequence bound to it. So you can see how the DNA is bound. You can see the unteasing of one of the strands that then forms the centerpiece for the generation of the RNA itself. It's the smallest RNA polymerase known. It's around about 100 kilodaltons in mass. I think it looks like it probably was at some point evolved from gene duplication. There looks like a center of symmetry there. There are no accessory proteins or cofactors. Everything is just driven by the triphosphate to monophosphate reaction of the nucleotides. There are several things happening all at once. So it needs to bind to the promoter on the DNA strand, separate the DNA strands, moving through a transcription bubble, which is probably about 13 or 14 bases in size, moves the template strand past the active site, incorporating the capping molecule at the start. It polymerizes the RNA nucleotides complementary to the template strand, and then detaches the DNA template at the end of the transcription, and then it starts again with another DNA strand. And it takes, we reckon, probably about 4 minutes for the polymerase to work its way down to form an RNA sequence from a DNA strand, certainly with the long sequences that you have for with self-amplifying RNA. Say something about capping. 5' capping is essential for eukaryote mRNA. Eukaryotes expect their mRNA to be capped, and this is the sort of capping structure they're expecting. With this guanyl group here, the mRNA comes off here. It's essential for translation and for avoiding being flagged up by the innate immune system. There are 2 ways of doing this. One was by post-transcription enzymic capping, which you can do using 4 enzymes: a triphosphatase, a guanyl transferase, a methyltransferase, and a ribotransferase. So you can do that all with 4 enzymes, but you need to find these 4 enzymes. The way we've adopted it, and the way that certainly a lot of other people, including BioNTech, have used, is this approach using a capping analog manufactured by TriLink Biotechnologies. Gives an authentic 5' cap end and has high capping efficiency. You just throw the capping reagent into the in vitro transcription reaction, and no need for a purification step. Moderna, we think, uses a modified form of this enzymic sequence using 2 enzymes which combine a couple of the steps shown here that have been derived from viral capping sequences. So capping is very important. In terms of purification, some sort of scheme like this is followed. The mRNA, say, is 4 to 12 kilonucleotides in size. That translates, certainly for the mRNA, the self-amplifying RNA, into a structure probably about, into sort of virus-sized territory, into things around about 4 or 5 megadalton. They are quite large particles, 30 to 50 nanometers in diameter. An unusual thing in bioprocessing with making mRNA is all the impurities are defined. You know the enzymes you've added to the mix. You know the polymerase has got a molecular weight of that, the DNA is that, the pyrophosphatase is that, and the RNase inhibitor is that. You know their charge, and you know their charge at different pHs. So it's possible to use a sort of rational design of purification process to separate these out. Other things that you have present are residual nucleotides and DNA fragments, excess capping reagent. So you can do a lot of purification by based on size differences and then known protein properties. So you've got your reaction. After that, you'd maybe do something like tangential flow filtration to remove the small molecule components, things like the nucleotides, maybe the DNA fragments. You'll clear out some of the smaller enzymes because they're going to be much smaller than the RNA itself. So by choosing the right size of membrane, you can manage to flush out a lot of the enzymes and most of the small molecules. Then by choosing a suitable chromatography chemistry, you can either bind the RNA or you can let the RNA flow through and bind all the impurities. A further round of tangential flow filtration to get the RNA into a suitable buffer for encapsulation. After encapsulation, you might have another set of tangential flow filtration to both concentrate and buffer exchange, ready to generate the final bulk product. And then one purification option is to make use of the base pairing between the adenosine on the poly A tail with a polythymidine tail that you would attach to a chromatography bead. And this is a well-known way of purifying mRNA. It's manufactured by several chromatography vendors. So this is some data on BIE separations, but people like Thermo Fisher or Cytiva manufacture their own oligo DT media. So you bind at high salt, which suppresses the charge on the phosphate groups so that the interactions are governed by base pairing. To elute the mRNA, you'd then remove the salt concentration, allowing the charge repulsion to take over from the base pairing, and you generate a nice chromatogram like this where you've non-RNA contaminants just flow straight through. You bound on your RNA at high concentration, and then you reduce the ionic strength and elute the mRNA. And it separates mRNA with a poly A tail, which is a good point because some of the polymerase may have dropped off early on before it got to the poly A tail. So this ensures that you're making an mRNA that contains the poly A tail. And then encapsulation of the RNA is essential for administration. So typically, and to date, all the methods of supplying mRNA into the body have used a lipid nanoparticle. And that's based on a complex lipid mix as seen here, which is the lipids used by BioNTech to make their mRNA vaccine. It's made of a non-ionizable lipid, which is this one here. It's positively charged at low pH, but neutral at physiological pH, and it will complex with the RNA. A pegylated lipid, which is shown here, which reduces antibody association, a phospholipid, and a cholesterol to stabilize the lipid nanoparticle. And these are in quite specific ratios, as you can see here, as this is described in the approval documents for the BioNTech vaccine. The bulk of the lipids are the cationic lipid to bind the RNA. There's then small amounts of the pegylated lipid and larger amounts of then the structural lipids like cholesterol, DSPC, to structure the nanoparticle. Nanoparticles are about 80 to 900 nanometers in diameter, so they can still be filtered through a 0.2 micron filter, and there's around about 100 RNA molecules per lipid nanoparticle. The nanoparticles are formed by mixing the aqueous RNA stream with the lipid mixture and ethanol. And the effectiveness of the lipid nanoparticle can be shown here. This is an agarose gel of some mRNA. So lane 2 is the mRNA itself, the intact mRNA. Lane 3 is the mRNA with the an RNAs added. You can see that you get the long RNA is being degraded into a mush of small RNA fragments. There's the mRNA in a lipid nanoparticle, and there's the lipid nanoparticle when presented with mRNA. It doesn't enter the nanoparticle. The mRNA is quite stable. Okay, so that's, I think, a little bit about background to mRNA itself and how you make it. In terms of supporting mRNA manufacture in the UK, at CPI we have established the mRNA Centre of Excellence. There's been constructed in Darlington adjacent to CPI Biologics. It's they've been pouring concrete this week. It's designed as a GMP facility for clinical and commercial production of mRNA. The artist's impression shows Darlington a very sunny day. It's not always sunny in Darlington, I have to say. And it's a manufacturing facility which will handle all stages of mRNA production through to bulk encapsulated mRNA. And it's aiming for use with vaccines and other mRNA applications, because as well as vaccines, it's we can see lots of applications for mRNA in gene therapy type roles. People have been using it for transient production of antibodies. People have been using it for delivery of CRISPR enzymes. And this facility should be completed in early 2022, early next year. So the final conclusions are it's RNA is a flexible and compact platform or gene delivery technology. It's well suited to rapid vaccine response. And as you can see, the first vaccine out there was an mRNA vaccine, and that just attributes or attests to the speed with which you can develop and produce these molecules using the technology. It's technology now validated by approved vaccine products, and as I say, it's applicable to a whole range of other therapeutic areas, from oncology through production of proteins within in situ to doing CRISPR type approaches. It's still very early in development of the technology, so there's significant scope for learning and innovation, and CPI's capabilities, expertise and facilities are well matched to develop the mRNA technology. As well as the Centre of Excellence for mRNA itself, we have a lot of prior knowledge in making lipid nanoparticles in our formulation group. And then some quick acknowledgements. There are too many to list them all, but acknowledge the work of the CPI mRNA team, both in biologics and formulation, and the work we did last year with Professor Robin Shattuck's sa-RNA group at Imperial College. And just thank you for your attention.